Hi, my name is Anael. I work at Oswaldo Cruz Foundation at Cellular Communication Laboratory. We study here P2 receptors and gap junction in the context of immune system and liver. And today I will present you the microinjection assay. Gap junctions play some physiological roles as synaptic transmission, heart contraction, differentiation, and others. And with this technique, we can study if the gap junctions are functional or not. Okay, let's see the basic components of the microinjection setup. Here we have the fluorescence, the inverted fluorescence microscope. When we will see the cells, this is a CCD camera to recording the results. We have here the current generator that will introduce the dye into the cell. Next, we have the holder when the microelectrode is connected, and the micromanipulator that's connected to the holder and allow the movement of the microelectrode in the three axes Z, Y, and X. Before start the experiment, we have to make the microelectrode. We use this borosilicate glass cap.